Hello, everyone. So today we are going to start with a second part of the cerebellum, and in this part two, we are going to discuss the internal circuitry, cerebellar cortex, the organization, and the cerebellar nuclei, and finally, we're going to look at the cerebellar pathways. So, cerebellar cortex, let's start with that. The majority of the neuronal cell bodies are located in the cerebellar cortex and they are further divided into three layers. The outermost layer is called the molecular layer, the middle layer is called the Purkinje cell layer, and the innermost layer is called the granular cell layer. So let's start with all of these layers, and we're going to go from outside to the inside. On the cerebellar cortex, the outermost layer is called the molecular layer. It is the outermost layer, and it is also the location of the stellate and the basket cells, which are important for the cortical interneurons. So over here, you're looking at the dissection or dissected part of the cerebellum. Uh, this one right here is the molecular layer. The yellow one is the Purkinje layer, Purkinje cells, and the last one is the granular cell layer. So you see here that you can only appreciate the basket cells over here. I cannot find an image for the basket for the stellate cells, but here you can see the basket cells. The second one is the Purkinje cell layer, and this is one of the most important cell layers for the cerebellum. So it is the middle layer, and it consists of two cell types: the Purkinje cells and the Bergman glia. So let's start with the Purkinje cells. So these are the largest cells in the cerebellar cortex, and they are arranged in monolayer. And their axon will advance into the cerebellar nuclei and the molecular layer. So molecular layer, as we discussed, is the outermost layer. So it starts from here and goes up. So we are looking at the Purkinje cell axon. So the cell body is right here. So it is going up and down. And it's mostly present right here. So the inhibitory and the GABAergic cells are basically present here, which inhibits the neurons in the deep cerebellar nuclei, and they only output neurons to the cerebellar cortex. So these are the only one that have the output in the cortex. Bergman glia, these are the specialized astrocytes and are closely associated with the Purkinje cells. So the most important one right here is the Purkinje cell. And if you remember from part one, the destruction of the Purkinje cells in the vermis is basically giving them patient is having a sway in the direction with their eyes open. So they have the ataxia of the trunk. And mostly present in the chronic alcoholics because that will cause the thiamine deficiency. The last one is the granular cell layer, which is the innermost layer. This green layer right here is the granular cell layer, and it consists of primarily two type of cells: granule cells and the Golgi cells. So the granule cells are the excitatory cells, and their axons are parallel fibers that stimulate the Purkinje cells, and the key neurotransmitter here is glutamate. The second one is the Golgi cells. These are the inhibitory cortical neurons, and their axons terminate on the dendrites of the granule cells. The main neurotransmitter here is the glycine. So you can see the Golgi cell right there, and you also appreciate the granule cells right there. So now we're going to discuss the interneurons of the cerebellum, as they play a very crucial role in the functioning of the cortical microcircuitry, and they provide the inhibitory input to the projections on the pyramidal neurons. So they are divided into two categories: inhibitory glycinergic cells and the inhibitory GABAergic cells. So let's start with the inhibitory glycinergic cells. They are from the granular layer. Major cells that are involved here are the Golgi cells, and they are primarily located in the granular cells, and their axon terminates on the dendrites of the granule cells. The second one is the inhibitory GABAergic cells. These include the cells from the molecular layer, and their major cells involved in these connections are the stellate cells and the basket cells. Molecular layer, if you remember, is the outermost layer, and both cells that are present over there, the stellate cells and the basket cells, are involved here. The stellate cells are located in the upper molecular layer, and their axon terminates on the dendrites of the Purkinje cells. And the basket cells are basically present in the deep molecular layer, and again, their axon terminates in the Purkinje cells. So now you look at the Purkinje cells is present in the middle layer or the Purkinje layer. So the Purkinje cells have the connections with both up and down in both layers. So now we're going to look at the cerebellar nuclei. So the areas which makes the cerebellar nuclei are basically paired. So that includes the nucleus dentatus, the nucleus interpositus, and that actually further consists of two nuclei, the nuclear emboliform and the nucleus globosus. And the last one is the nucleus fastigiae. So let's start with the nucleus dentatus. In this image, you can appreciate that the dentate nucleus or nucleus dentatus, you can say it both ways, are basically paired and they are present in the most lateral part. And so these are the most lateral of the nuclei. So their efferent fibers, the fibers that they give out from here, are sent to the ventral anterior nucleus and ventral lateral nucleus of the thalamus. The second one is the nucleus interpositus. So interpositus is basically made up of small nuclei right here, and they have two nuclei. One is the nucleus emboliform, these ones right here, and then we have the nucleus globosus or the globose nucleus, these two present right here. The nucleus emboliform, it is located medial to the nucleus dentatus. Obviously, if you look at the image, this is the dentate nucleus, and the emboliform nucleus is more medial, and nucleus globosus is further medial to the nucleus emboliform. So you go from lateral to medial, you'll find dentate nucleus, emboliform nucleus, globose nucleus, and the efferent fibers, nerve fibers from these nuclei will connect with the red nucleus of the midbrain and the reticular formation. The final cerebellar nuclei that we are going to discuss is the nucleus fastigiae or fastigial nucleus, and these are present most medially. So these two purple circles that you look here in the image is the fastigial nucleus. Their input is primarily from the inhibition from Purkinje cells and excitation from the mossy and the climbing fibers, and their output or the efferent fibers are to the vestibular nucleus, and their primary output is to the glutamatergic relay neurons. So now we're going to look at the cerebellar connections. So if you remember from the first part, there's three peduncles that are going to connect the cerebellum with the midbrain. So the first one is the superior cerebellar peduncle, middle cerebellar peduncle, and the inferior cerebellar peduncle. So there's two type of pathways. One is the cerebellar afferent pathways, and then the second one is the cerebellar efferent pathways. So let's start with the afferent pathways. So all the afferent pathways or all the afferent fibers, they pass through all three cerebellar peduncles and goes to the cerebellar cortex. So there are two extracerebellar excitatory glutamatergic efferent system. One is the mossy fibers. So they originates in the spinal cord and brain stems, and they synapse on the Purkinje cells. You remember the middle layer of the cerebellum. Purkinje cells are present there. So the mossy fibers are going to originate in the spinal cord and gonna go up and connect with the Purkinje cells. The second one is the climbing fibers. So they originate in the contralateral inferior olivary nucleus within the medulla and synapse on the Purkinje cells. Again, mossy fibers, climbing fibers, both synapse with the Purkinje cells. So now you can understand the importance of the Purkinje cells present in the cerebellum. So you see the mossy fibers, this one going here, and this is the climbing fibers that is going right there. You see the pontine mossy fibers also going in there from there. So this is important. So we're going to look at the cerebellar efferent pathway. So these pathways of the cerebellum, they pass from the cerebellar nuclei to the following structures. So which structures does it connect with? So these are the efferent. So these nerves originate in the cerebellum and they leave through the peduncles, and then they connect with thalamus, vestibular nuclei, red nucleus, and the reticular formation. So you look at the pons, this is pons right here. We have the corticopontine fibers that making synapse, and the pontine fibers, mossy fibers actually entering from the middle cerebellar peduncle, making its connection. So you have fastigial nucleus interposed in dentate nucleus. So dentate nucleus, efferent fibers relieving from the superior cerebellar peduncle to thalamus and the red nucleus. We have the interposed nucleus efferent fibers relieving from the superior cerebellar peduncle to thalamus, and then we have the fastigial nuclei. So In the previous part, we have already discussed that based on connections, cerebellum is divided into three regions. We have the vestibulocerebellum, which is present in the midline. Then we have the spinocerebellum, and then we have the pontocerebellums. They were already discussed in the first part, so please check the first part and familiarize yourself with the afferent and efferent fibers that enter into these regions and leaving from there. With this slide, we're going to end our cerebellum, and in the next couple and the next presentations, we're also going to discuss further parts of the brain. Thank you so much for joining, and I'll see you soon.